Welcome to Unit 3. We are going to start off by talking about cells. Again, here's a helpful link. And let's begin. So, human cells really have three main parts. We are going to spend a significant amount of time in the second part of Unit 3 talking about the first one, which is the plasma membrane. Plasma membrane is the outer boundary, and everything else within the cell is going to be found within. That inner portion is divided into cytoplasm and the nucleus. The nucleus is also membrane bound. It is its own separate, distinct pocket within the cell. It's going to control all of the cellular activity. The cytoplasm is everything else. Cytoplasm as a term is going to refer to both the fluid within the cell as well as all of the organelles within the cell. If we are just talking about the fluid within the cell, we're going to refer to it as cytosol. Here is an artistic rendering of a cell, specifically an animal cell. When we go over these cell parts, these organelles, I do need to have you just keep in the back of your mind the fact that not every cell is going to have all of these parts. What organelles a cell has is dependent upon its function, what its role is within the human body. For instance, your fat cells do not have mitochondria. Why don't they have mitochondria? Well, they don't create ATP. On the other hand, your muscle cells need and create a ton of ATP and therefore are going to have a lot of mitochondria. All right, so there's this thing called modern cell theory. Modern cell theory is the thought that all organisms are composed of cells and cell products, organelles. Cells are considered to be the simplest structural and functional unit of life, as we saw in our organization chart back in Unit 1. And we do not really have anything smaller than a cell that we consider alive. Now you might be thinking in your head, well, what about a virus? Viruses, there's a lot of hot debate on whether or not we consider them alive. You might think prions, if you've heard of them. Prions are just proteins, abnormally structured, put together proteins, but they're just proteins. They have no characteristics of life. Now, that being said, cells can get pretty small. So just because cell is the smallest, simplest, does not mean that it is large. As I mentioned, structure and function are going to dictate the activity of the cell and therefore what is in it. Cells come from pre-existing cells. Life begets life. Life is not created from a rock, things that are non-living. Because of this, all life is going to trace its ancestry through the same original cells. We have yet to find anything that refutes this theory, and that is where really the class zoology and the study of zoology comes into play, is tracing that ancestry. Because of that common ancestry, all cells of all species will have basically the same similarities in chemical composition, metabolic mechanisms, structure, the whole nine yards. If that is something you are interested in, really wanting to understand, I do encourage you to take zoology if you have not. If you have room for another elective, it's a really good one. It really gets into those fundamental similarities and how they've progressed through the various stages as life became more and more and more complex. All right, so brief intro on the cell membrane. It's also referred to as a plasma membrane. As I mentioned, it is the boundary, keeps everything that's supposed to be in, and everything that's supposed to be out, out. It's also going to control the interaction with other cells. How exactly that happens, we'll talk about later in this unit. It's going to control the passage of materials in and out. Again, we'll talk about how later. And when viewed on an electron microscope, this is going to be our extracellular. This is going to be our intracellular. Extracellular is referring to the side of the cytoplasm that faces out of the outer side world of the cell. Intracellular is facing the cytoplasm, everything that is inside the cell. Now, what makes up a plasma membrane, how it works, how it functions, has gone through a lot of iterations through the past couple hundred years as our knowledge and technology has expanded. Currently, we know that it is an oily film of phospholipids that have diverse proteins and carbohydrates and structures embedded within it. Again, we'll get into more detail in that in a bit. But this is an artistic rendering of what that may look like. Okay, cytoplasm, everything inside the cell. Now, the organelles found in the cytoplasm are divided into two categories: membrane-bound and non-membrane-bound. It sounds exactly like what it is. Membrane-bound organelles have a phospholipid membrane that surrounds them. These are things like the nucleus, mitochondria, lysosomes, peroxisomes, endoplasmic reticulum, both smooth and rough, and Golgi apparatus. Non-membrane-bound organelles are, again, they do not have a membrane surrounding and containing them, so they are really one with the fluid. These are things like ribosomes, centrosomes, centrioles, basal bodies. Now, those are the two things that people tend to talk about, the fluid and the organelles. But there are other things found within a cell's cytoplasm. This is going to include things like the cytoskeleton, which is a collection of microfilaments and microtubules that lend support and structure and aid in the transport of things around the cell. We'll talk more about them. And things that are called inclusions. Inclusions are stored products. Let's start off with the nucleus. Here's the thing. Every cell either has or had a nucleus. Now you might be thinking to yourself, wait, we have cells that don't have a nucleus. Yes, your red blood cells. Your red blood cells, in their process of becoming a red blood cell, lose their nucleus, which means they lose their ability to self-replicate. They actually are dependent upon, they're called precursor cells, but basically think of them as stem cells. So it's these stem cells that divide and differentiate themselves, creating red blood cells versus the red blood cells themselves. The red blood cell loses the nucleus. But at one point they did have them. So, as far as we have been able to tell, the largest organelle in a cell is the nucleus. It's about 5 micrometers in diameter. Some, as I mentioned, no nucleus, and some are what we refer to as multinucleated, meaning they appear to have more than one. A great example of that would be a variety of our white blood cells. The nucleus is surrounded by a nuclear envelope, also referred to as the nuclear membrane. This is a phospholipid membrane that's held together and has what we call nuclear pores. Nuclear pores are what enable substances, including water, to move in and out of the nucleus. Now, located within that nuclear membrane, we are going to find nucleoplasm. What is nucleoplasm? Nucleoplasm is going to be fluid plus everything found within the nucleus. If you're going, wait, that sounds like cytoplasm, you would be correct. It's that same concept. Specifically, though, the nucleus is going to have chromatin. Chromatin is a thread-like matter containing DNA and protein. This chromatin condenses down and becomes chromosomes during the replication process. We're also going to find the nucleoli if it has more than one, or a nucleolus if it only has one. The nucleoli or nucleolus is a dark mass located within the nucleus, which is where ribosomes are produced. All right, electrons microscope image. Chromosomes, chromatin that have condensed down into chromosomes. Right. Moving to our next organelle, endoplasmic reticulum. The endoplasmic reticulum is divided into two categories, rough and smooth, and that is based upon its appearance. Rough ER is rough in appearance because it has an organelle called ribosomes embedded within its membranes, giving it a rough appearance. Rough ER is going to be parallel units of membranes with folds and pockets within them. They are going to run continuous with the nuclear envelope, meaning they're going to run right into each other. And rough ER is going to be involved in protein synthesis and production of other cell membranes. Rough ER is going to run continuous with smooth ER if the cell has smooth ER. Smooth ER is smooth in appearance because it does not have ribosomes embedded in it. Therefore, it is actually not going to be involved in protein synthesis. It's going to be more tubular and branching in appearance. And it's actually going to be involved with lipid synthesis, detoxification, and calcium storage. Here's an artistic rendering of what that looks like. This structure right here is the Golgi apparatus, and that's what we're going to talk about next. The Golgi apparatus, or complex, depending on the textbook, is where those lipids and proteins that are produced by the smooth ER are packaged and processed. So they're cleaned up excess amino acids, uncoded or unnecessary amino acids and DNA, RNA are removed, and they're cleaned up and put in pouches to go wherever they need to go. The Golgi apparatus is going to package them up in what we call Golgi vesicles, which are just irregular sacs that bud off. Some will become what we refer to as lysosomes, which lysosomes we'll talk about here shortly. Some are going to fuse with the plasma membrane, and some are going to become what we call secretory vesicles. And those that fuse with plasma membrane and secretory vesicles are exactly what they sound like. They're going to enable the removal and of substances into and out of the cell. Specifically, secretory vesicles are going to store it for a later release. So it's a very temporary storage. Right. Within cells, we have pouches called vacuoles. Vacuoles are pouches that store nutrients and contain enzymes, depending on the type of vacuole, to degrade waste products. Lysosomes, specifically, are going to be packages of enzymes in a single unit membrane, meaning it's one layer, varies in shape, because it depends on what it has within it. If it is involved in intracellular digestion, it will have enzymes that will help hydrolyze proteins, nucleic acids, complex carbohydrates, phospholipids, and any other substance within the cell. If it has enzymes that are going to be involved with autophagy, that is going to be enzymes that enable it to digest worn out and unnecessary organelles in mitochondria. Some lysosomes will have enzymes involved with autolysis, which is also known as apoptosis or programmed cell death. And some will have enzymes involved with glucose mobilization. These ones tend to be found in the liver and help cells break down glycogen into usable glucose molecules. So what a cell has and what enzymes are in these lysosomes all depends on function. An example of how they would work. Now, peroxisomes are similar to, but slightly different from lysosomes. One way in which they are different, they're not produced by the Golgi apparatus. However, they are typically found in all cells. They are pouches, they are a single membrane, and they do contain enzymes. However, peroxisomes contain a very special batch of enzymes. These enzymes are designed to break down H2O2, or hydrogen peroxide. Why does this matter? Hydrogen peroxide is a poison. It will kill a cell. So, in order to get rid of hydrogen peroxide so it cannot kill the cell, we need to have an ability to break it down. Why does this matter? We don't drink hydrogen peroxide. That would be true. We don't drink hydrogen peroxide. However, hydrogen peroxide is created as a byproduct of a bunch of natural processes that our cells do in order to survive. So it's created as a byproduct. That byproduct needs to be gotten rid of, hence peroxisomes. Now, the liver and the kidney have way more peroxisomes than any other cell in the body, or cells in the body, I should say. And that's because peroxisomes, because they enable the breakdown of hydrogen peroxide, are going to be involved in the process of alcohol detoxing and killing bacteria. Ribosomes appear as small dark granules of protein and RNA. They're a complex of RNA and protein, and they can be free floating in cytosol or found on the surface of rough ER, as we've mentioned. What is their purpose? When we talk about reading DNA, there is a strand of RNA called messenger RNA. And that strand of messenger RNA is going to end up going to a ribosome and getting interpreted through a couple more steps into a chain of amino acids. And it's the ribosomes that are going to do that reading and connect the amino acids together. Mitochondria. Mitochondria are a double-layer membrane organelle. That inner membrane is going to have folds, which are called cristae. The mitochondria is also referred to as the powerhouse of the cell, and it is going to be involved in the creation of ATP. If you do not have mitochondria in a cell, the amount of ATP that can be created is limited. And this is just due to the energy production process. Here's the thing. Mitochondria are special. Mitochondria, unlike any other cell organelle in a cell, have their own DNA. They have a small circular DNA that's called mitochondrial DNA and looks very similar to a bacterial plasmid, for those of you who have taken microbiology. Also, the mitochondria contains ribosomes. So there's a theory that mitochondria were once actually bacteria that took up a symbiotic relationship with another cell, another bacteria or a eukaryotic cell, and they merged together because of that symbiotic relationship. I haven't seen anything recently, not that I've looked, proving that, but it is a current theory. One of the things that helps support this theory is because mitochondria can reproduce independently of the cell. The cell does not need to undergo its cell reproduction cycle in order to make more mitochondria. The demand for energy increases, it can create more ATP or more mitochondria to make more ATP all on its own. And mitochondria can survive on their own for 10 days. Very, very unique. Here's an electron microscope image of that along with our artistic rendering. All right, that cytoskeleton that I mentioned. It is a collection of filaments and tubules that provide internal support and movement, as I mentioned. What are these filaments and tubules? Well, microfilaments, these are going to be strands of actin, which is a protein complex. They're going to form what's called the membrane skeleton. These are going to support phospholipids, support microvilli, which are finger-like projections of the cytoplasm. They're going to, if the cell moves, produce that movement, and when combined with myosin, which is another protein chain, they're going to be what causes your muscles to contract. We'll talk about that more later in the muscles, as well as in physiology. If we're talking intermediate fibers, these are going to be fibers that form junctions that hold your epithelial cells together and resist stresses on a cell. They are usually going to form what are called tight junctions, which help keep the cells together and withstand those stresses that would pull them apart. We also have microtubules, which are going to be just what they sound like. They're going to help maintain the cell shape and move organelles within. They're also going to be found within structures called cilia and flagella, which we'll talk about in a moment. All right. Microtubules are cylinders of 13 parallel strands of a globular protein called tubulin. This tubulin forms a long chain that eventually becomes called a protofilament. These microtubules are going to form tracks. Think of a railroad, very similar in nature. They are also going to be involved with giving structure to cilia and flagella and the centrioles and mitotic spindle involved with the cell cycle. They can be permanent, but they can also be temporary. For instance, found within cilia and flagella, permanent. Found within a mitotic spindle, temporary. They're assembled and disassembled as they are needed or not needed. Imaging. All right, what is a centriole? A centriole is a short cylindrical assembly of microtubules arranged in 9 groups of 3 tubes each. You have 2 centrioles. They typically hang out perpendicular to each other. These centrioles are going to hang out right near the nucleus, and they're really not going to play a role until the cell is undergoing a reproduction stage. So it's starting to divide and split. If we are involved in cilia formation, a centriole is going to migrate to the plasma membrane. 2 microtubules will elongate to form the microtubules that will create the cilia. This will develop in usually less than an hour. What are cilia? Well, cilia are hair-like processes, about 7 to 10 micrometers long, single, non-motile, meaning they don't move anything, they don't move at all themselves, are found on nearly every cell. They're like a hangover type feature. However, on many cells found in lining the respiratory tract or lining your uterus, fallopian tubes, any place where we have mucous membranes, we are going to find these cells are covered in upwards of 200 on a single cell. And the reason for this is they are going to move in a wave-like motion to move substances, specifically mucus, over them. Now in the respiratory tract, that's going to help move mucus, which has trapped gunk in them, like dust, debris, bacteria, up and out of the lungs so it can be taken care of and no longer harm the lungs. If these cilia are found in the inner ear or in the retinas, yes, the retinas have cilia. If they're found in the nasal cavity, they can also be found to be part of the sensory complex. Sensory complex how? Sense of hearing, sense of smell,